Hello, welcome to Goal 3, Cells, Module 2. Let's get inside. The objectives of this module are to identify the organelles and describe their functions, and talk a little bit about cellular respiration. The cytoskeleton is like the framework that keeps cells in a 3D shape. Think of a cell like a ball. What keeps the ball inflated? Air pressure for a ball, but in cells the ball is made of a lipid membrane and there's fluid inside. So to keep it from collapsing or smushing, it has a scaffolding system inside made of these fibers. There are a couple of main fiber types. Microfilaments, like actin, give strength and shape. Intermediate filaments are the strongest and most durable, and they're also used to move things inside cells. Thick filaments like myosin are only found in muscle cells, and microtubules give strength but are like train tracks inside cells and move things, including the centrioles, which are going to be involved in producing the spindle during mitosis. The endoplasmic reticulum is a membrane-like group of sacs that's continuous from the nucleus. It's made up of cell membranes, so phospholipids, and there's ribosomes attached. Ribosomes are where proteins are made, and as they're made, they're put into the endoplasmic reticulum and then are transported to wherever they need to go in the cell. Ribosomes are made out of RNA and protein, and if ribosomes are attached to the endoplasmic reticulum, it's called the rough endoplasmic reticulum. And if they're not attached, then those areas of the endoplasmic reticulum without ribosomes are referred to as the smooth endoplasmic reticulum. So the endoplasmic reticulum is kind of your transportation highway system, and the ribosomes are where proteins are made. The Golgi apparatus, you can think of this as FedEx. It's also made up of sacs called cisternae, and it's kind of between the endoplasmic reticulum and the outside, because if proteins are made on the ribosomes, put into the endoplasmic reticulum, transported to the Golgi, and the Golgi, they're packaged for secretion. They're packaged in a little membrane encircling the secretion. It's called a vesicle, and it is released from the Golgi, and it merges with the cell membrane, and excretion happens. So here's a picture of a little particle. You can see it traveling the endoplasmic reticulum, and it goes over to the Golgi, and the Golgi kind of preps it and forms a vesicle, and as it travels through the Golgi, it gets packaged, and soon it will go out of the Golgi and merge with the cell membrane, which is this blue thing down here, and that particle is secreted. Peroxisomes contain a whole bunch of enzymes which primarily function together to get rid of toxic substances, particularly hydrogen peroxide. This is a common byproduct of cellular metabolism, and it's quite dangerous. So these organelles have enzymes that convert it into water, rendering it, pretty much safe at that point. Some peroxisomes also help break down long-chain fatty acids and detoxify alcohol, particularly those in the liver. And as those are detoxified, they become less harmful to the human, obviously, and sometimes oxygen is actually produced. Others are involved in making phospholipids, which of course you need for new cell membranes. So you can think of peroxisome as the detox center. Lysosomes are like the garbage disposals of the cell. They get rid of unwanted material, usually inside is slightly acidic. There's over 40 enzymes in a lysosome. There's lots of these in white blood cells, because white blood cells' job is to kind of go around and clean up the mess, so they will engulf or phagocytose large things like bacteria. Once inside, the bacteria are put into the lysosomes or merged with the lysosomes, and the acid chews them up, as well as the enzymes. Tay-Sachs disease is actually due to an abnormal lysozyme enzyme. So here again you can see a picture of a cell, endoplasmic reticulum. It's rough here, it's got the ribosomes attached. The Golgi is FedEx. The lysosome is the garbage disposal. It took in some of this stuff and digested it. So hopefully you can see some of the different organelles and how they might be connected there. The mitochondria is where energy is made. Mitochondria are little sacs, and inside is another set of membranes, so you have an inner and an outer area inside the mitochondria. Cells that have more mitochondria can produce more energy. In fact, the more you exercise, the more mitochondria your muscle cells will have. These organelles are sometimes called the power packs of the cell because of the energy that they generate. Inside of the mitochondria, those inner membranes, it contains a matrix, fluid-filled matrix, and that is where the ATP is made. Interesting, mitochondria have a little bit of their own DNA. It is separate from the nuclear DNA, and you got it all from just your mother, because only eggs, not sperm, have mitochondria when they merge with each other to form a new human being. So all your mitochondrial DNA came from mom. Now, mitochondria do a process called cellular respiration. It is the way cells make energy, and essentially it's breaking down sugar to generate energy and some waste products like carbon dioxide, heat, and water. And to do it, you need oxygen. There are 3 steps to cellular respiration, and the equation is down here at the bottom, and to go through the entire equation you need all 3 of these steps: glycolysis, the Krebs cycle, and the electron transport system. But what you're doing is taking glucose, C6H12O6, in the presence of oxygen, and you're generating water, carbon dioxide, and ATP. And ATP is the form of energy that cells use. Cars use gas, cells use ATP. Okay, now glycolysis is the first step, and you only get 2 ATP from that step, so you don't get a lot of energy from it, but that step doesn't need oxygen. However, we can't live on just step 1. Glycolysis is not enough energy generating process to give us more than 4 minutes. So that's why I say you need oxygen, because without it, even though you can do step 1, you cannot get enough ATP to live. So here's the mitochondria, and glycolysis takes place just outside the mitochondria in the cytoplasm, and basically you go from glucose to pyruvate, or pyruvic acid, and from that you get just a little bit of ATP too. Not enough to live on, but you do get a little. The pyruvate is taken into the mitochondria and converted to acetyl-CoA as it goes in. And it goes through process number 2, which is the Krebs cycle. And as it kind of goes around this circular process, you can get more information about this in a bit here, it clips off the carbons. And remember, carbons are covalently bonded molecules, and as you clip them off, when you break a covalent bond, you get a little bit of energy. So you get a whole lot more ATP, and you get some NADH. NADH is basically a suitcase carrying electrons. You got one from glycolysis, and you get a whole bunch more from the Krebs cycle. These electrons are put into step 3, the electron transport chain. And as the electrons move through the different molecules in the electron transport chain, you generate a whole lot of ATP. Now, the carbon dioxide that is made during the Krebs cycle is released into the cytoplasm and then diffuses across into the blood, where it's taken to the lungs and we exhale it. There is a little bit of water created here, but of course water is not harmful. And besides ATP, you also get a little bit of heat. And even though it dissipates and sometimes it's called waste, we use it to help maintain body temperature. Okay. So you should stop here and you should click on this link, because this tells you all about cellular respiration done by the Amoeba Sisters, which is an awesome set of YouTube videos. So please use this link and listen about cellular respiration. They can do it way better than I can. We'll also talk about this in class. So use your lab book to help with identifying the organelles. You can also type in any organelle into Google and get more information, as well as ask questions.